I'm now going to explain to you in slightly more detail how the human body, or let's say the mammals in generally, how they produce antibodies, okay? And the antibody production is a pretty complicated system or a complicated procedure because we gotta really make sure that we should produce antibodies only when you really need them, okay? And we don't just want to make antibodies for nothing. And the first thing is you have to understand there are a whole bunch of white blood cells. So far we only talked about white blood cells, but you have to understand there are many subcategories of white blood cells, all with different tasks. And the first step in antibody production is the activation of so-called T helper cells, okay? That's their name. How do we activate them? And first of all, we need to have an antigen. Let's say we have a bacterial, some kind of an infection, and I don't know, I'm going the virus or bacterium, and that is my my pathogen. Okay, and you've got I don't know where you caught that. You've got an infection somewhere, and this pathogen is now eaten up by macrophages. So the first thing that we need is we have this macrophage. How am I going to draw this? Okay. That is a white blood cell, okay, that's a macrophage, that's the nucleus, okay, and this pathogen is now eaten up, and the macrophage eats up because it flows around the pathogen, right, okay, that's the pathogen, and then it ends up in a vacuole, in a food vacuole over here, right, so that it's kind of eaten up, right, and now what happens next is it's going to be digested, and the pathogen, the pathogen is taken apart, and it's digested, broken down, and the macrophage, that's my macrophage, now takes the pathogen apart and presents on the outside, it's drawn too big, okay, but it will present antigens of the pathogen. So antigens are presented, and this one over here, this is a protein. It looks like an antibody, and it presents the antigen, okay. So that is the thing over here. It sticks out. It kind of tells the world, if you want to say, look, I found something. I caught a pathogen. Okay, and that's the antigen of the pathogen. Okay, look what I found. Look what I got. We've got a problem over here. We're being invaded by pathogens. A pathogen is a disease-causing organism. Okay. And then what happens next is this macrophage now wants to alert and warn other white blood cells, the so-called the T helper cells, okay? So the macrophage, that's not a T helper cell, okay? And what it does is now the following. I will now draw over here, I will draw my, I don't know, my T helper cell, okay? It's an inactive T helper cell, so I would say T sub H. And this T helper cell now also has these surface structures over here, and what the macrophage now does is the following. It will present the antigen that it caught, okay, it will present it like this to the T helper cell. That's my macrophage, okay, and then the T helper cell now senses, there is an antigen over here, okay, and the macrophage is activating it, and what happens now is it becomes activated. I'm just going to write like this, activated, okay, it turns on, certain DNA portions are turned on, and now the T helper cell which was floating around passively in the past, is now activated. Okay, so that's stage number one. It's the activation of T helper cells, and they are activated by macrophages. Activated by macrophages that in turn have been activated by an antigen. Okay, with antigen. Okay, that's step number one. Okay, and the T helper cells are now active and are now able to yet alert another cell, okay? And now what happens is the following. The B helper cells are now activated. B helper cells activated by the active T helper cell. This one over here. Okay, now how does this work? Well, surprisingly similar. You have over here an inactive B cell. Okay, that's the B cell. It's the inactive one, and this inactive B cell also has over here a surface structure that's an antibody which is actually on the surface. Normally antibodies are soluble, okay, and they're floating around, but here we have also these surface proteins, these antibodies sticking on the surface. And what happens now is the following. There are the antigens from the pathogen are also floating around in the blood, okay, and they bind here again, and now it binds here. So that is one way of activating it. That's an antigen, Ag antigen. Is we've got an infection, so there are plenty of these pathogens and antigens floating around, okay. And then the next thing is now we're also activating the whole thing by the active T helper cell over here, okay. Yeah, so that's the active T helper cell, Th active. If you want, yeah, that's the one over here, right? Okay, and that's a B cell, and now this activates the B cell. It becomes active. So what we have is the following. So, from the beginning, we have a macrophage. The macrophage activated the T helper cell. The T helper cell activated the B cell. Okay, and now as a next step, the B cell is now activated. Now, how does this happen? If this happens, if this binds, and actually, how does it activate it? It's because it releases chemicals. Okay. It activated by chemicals. Okay, so you see that it's got to be activated by the antigen binding and also by chemicals. Massively complicated process. Doesn't matter. We've now got the B cell, an active B cell, and what does the active B cell now do? The active B cell. Okay, where is this? The active B cell. Okay, will become a plasma cell. Okay, so the B cell forms. Not forms, or becomes would be better, becomes a plasma cell, and the plasma cells, they make Ig, immunoglobulins. It's the same as the antibodies, okay? So that is the thing. So we've got over here an active B cell, and this active B cell will then become a plasma cell. And the plasma cells, they have a lot, of course, they've got a nucleus over here, and you know what, they have a large amount of RER, rough endoplasmic reticulum. This is supposed to be the ribosomes, okay? Why? Because the antibodies are made on the RER, okay? Antibodies produced, and so these are supposed to be my antibodies which are produced over here, okay? And these antibodies then basically inactivate the antigen, okay? And we're not finished yet, because after the infection has been removed, okay, some of these plasma cells become memory cells, and they give long-lasting immunity. Long-lasting immunity. My handwriting is really bad. Immunity. In case for those of you who have a problem here, okay? They give long-lasting immunity because these memory cells, when there is later on the same antigen invading the body, these memory cells can form directly plasma cells. You don't have to go through this complicated activation process anymore. Memory cells can directly become antibody-producing plasma cells. And that is the reason why we have vaccinations, because we want to form these memory cells, because sometimes they can give us immunity for years. And when we get really infected by the same antigen, then they form plasma cells directly, much faster. Okay, and we don't have to wait several days for the whole activation of the T helper cell and the activation of the B cell, and then the B cell becomes a plasma cell. It takes a long time, okay? And in the future, however, the immune system remembers this by forming these so-called memory cells, and they can convert directly into plasma cells if they are exposed again to the same antigen, okay? So that was basically it. So maybe I should summarize this a little bit over here. Go back here. We have the macrophage, the T helper cell, the B cell. I just continue drawing over here. The B cell becomes, activates. The T helper cell activates the B cell becomes a plasma cell. Okay, and then which produces the antibodies, and then later on the plasma cell becomes a memory cell. and the memory cell can very quickly go back again to become a plasma cell. Okay, if there is another invasion with the same antigen. Okay, so that is kind of the process. Would be kind of nice to remember these stages here. Okay, so that's it. Okay, all the best.